Hey everyone, welcome to a cool episode of BioLogic Science News. Today I want to talk about the research on hair loss conducted by Choi Kang-yeol and his associates of Yonsei University in Seoul, South Korea. Their research has produced a groundbreaking chemical product that's been demonstrated to regrow hair. In the course of their research, they found that hair lost through stress or age isn't gone forever. The regrowth of that hair is possible thanks to the WNT beta-catenin pathway. This is a chemical pathway in the cells that's been associated with hair follicle development and hair follicle regeneration. Choi and his team of researchers found that there's an inhibitory protein, a CXXC type zinc finger protein called ZXXC5, that comes in and disrupts the WNT beta-catenin pathway. It does this by interacting with a protein in the pathway called disheveled, which is a protein free-floating in the cell cytoplasm, with a phosphate group attached to it that it can use to phosphorylate other proteins. Disheveled being able to phosphorylate other proteins it's involved in the WNT beta-catenin pathway, but the CXXC5 protein comes in and binds to disheveled and prevents it from interacting with all of the other proteins in the pathway that it needs to interact with. This disruption by CXXC5 prevents hair follicles from regrowing. The CXXC5 protein is present in normal cells with hair follicles, and it's one of the things that's responsible for hair loss in the first place, and the frustrating difficulty in trying to grow it back. Choi and his team found that by using a competing protein that binds to the CXXC5, they can prevent it from binding with the disheveled protein, and thus prevent it from disrupting the WNT beta-catenin pathway in the first place. They tested mice who don't have the genes for CXXC5, and thus they don't express it. These mice showed better than normal hair regrowth compared to mice that did have the CXXC5 genes, and when they were treated with valproic acid, they experienced even better hair regrowth. This chemical, valproic acid, is a glycogen synthase kinase 3-beta inhibitor that actually stimulates the WNT beta-catenin pathway. So Choi Kang-yeol and his research team found that CXXC5 inhibits hair regeneration, but that its effect can be suppressed with a competing protein. And they also found that valproic acid can be used to stimulate the regeneration pathway to compensate for any residual CXXC5 reactions. Using both methods can create a cellular environment very conducive for hair regrowth. Summarizing his research, Choi was quoted as saying, "We have found a protein that controls the hair growth and developed a new substance that promotes hair regeneration by controlling the function of the protein. We expect that the newly developed substance will contribute to the development of a drug that not only treats hair loss, but also regenerates damaged skin tissues." The research team called their competing protein PTD-DMB, and they tested it on lab mice to find that within 28 days, it really did stimulate the growth of new hair follicles and new hair. When PTD-DMB was used in combination with valproic acid, the regrowth of hair was accelerated. For people who are balding, or who have experienced burns on skin that usually has hair, like the scalp, this kind of research is really exciting because it's really promising. The results so far have been really effective, so Choi and his team expect their research to result in new drugs for treating hair loss and for stimulating hair growth in damaged tissues.